Whenever I will try to focus some videos. If it is not clear to you, please let me know in between the class. Can you see the slide now? Culture media and pure culture of bacteria.
No, sir.
It is a normal slide that I'm sharing. Oh, okay, maybe. Let me try once again. Oh yeah, share. I think now I can see. I guess now you can see the slide. Culture media and pure culture. Okay, fine. Thank you. So in the last couple of classes, what we are discussing about the basic techniques that we use in a microbiological laboratory for handling and identification of organism, and our main focus is towards the study of bacteria. So gradually we'll shift it to fungi and then to the viruses at a later part. So in today's class, what I'm going to highlight is on how we can propagate organism. This is quite the most interesting what I feel, and it's really an exciting area through which these small unseen organisms we can propagate and we can study them. We can propagate and enlarge their population to million folds, and then we can study their behavior, and we can crack the logic behind like how they can associate it with disease condition. Of course, the disease causing organism, individual organisms and their behavior we are going to discuss in later part. Today's class will be the most basic procedure that we follow for propagating the organism. So before going for, or on one hand, I can say it is something like a recipe for the bacteria. How we can cook the food for them so that they will be happy and they will be growing in our culture plate, and we can study them. So this is for the culture media. Some area already we discussed in the theory, but still I would like to recollect those informations to understand this chapter. First thing is that we know majority, almost to say 98 to 99% of the bacteria of our interest, they are heterotrophic organisms. Okay, so when we say heterotrophic, that indicates that means that these organisms can use the reduced organic molecules such as the sugar, amino acid, fatty acid, etc. Those macromolecules and the breakdown building blocks they can utilize as a source of carbon and energy. And utilizing this, they can go for biosynthesis and they can reproduce to their progeny generation. So, and we call them as the heterotrophic. At the same time, we also talk about the phototrophs, like which need the light as a source of energy. But majority of organisms, most 10% of the organisms, they are not phototrophics. They are heterotrophic. They only need the reduced organic molecules as a source of carbon and energy. So as a result, whenever we cook or we design any dishes for them or the culture media through which they can utilize their nutrient and grow and multiply, they must contain all these organic molecules. So considering it, what we know is that these nutrients in the form of water, like water, as you know, it's a major part of living creatures. We must provide the water to the culture media, and then the culture media must contain some source of energy. And beside the phototrophs is the chemotrophs, or organisms. They use the chemical compound as a source of energy, not the light. Then they need the sources of electron to carry out their all metabolic activity. So our organisms, heterotrophs, are also called as the organotrophs. They use the organic material as a source of electrons. Whereas there are certain bacteria, very small percentage, which are also called as the lithotrophs. They use inorganic salts as a source of carbon and energy. So likewise, these classifications we discussed in the theory: source of carbon, nitrogens, and the minerals, and of course they are living creatures. They also need the vital amines for their growth and multiplications. And finally, this is, although we keep it at last, but this is quite important, the gaseous requirement. On that basis, you have learned about facultatively anaerobic, aerobic, strictly anaerobic, and strictly anaerobic. These are the different classifications on the basis of gaseous requirement. As you know, there are certain organisms, such as certain bacteria, which cannot tolerate even the slightest concentration of oxygen. So we call them as the strictly anaerobic bacteria. So to grow those bacteria, you must provide an environment which doesn't have any single molecule of oxygen. So you have to create an anaerobic conditions for during the incubation of those organisms. So depending on their needs and nature, we have to provide the different conditions. So these are some of the basic things. So coming to the culture media, what we mean by culture media means this is a recipe, or it will contain all the essential nutrients as well as some non-essential nutrients for support the growth of the bacteria, so that they are happy with the media that you provide, and they will grow and multiply, and they will form a colony. Colony means, as you know, the bacteria, they reproduce by transverse binary fission. One bacteria, they will grow up in the lag phase of their growth curve, and then when the log phase starts, then from one bacteria, two bacteriums are produced. The septum was produced. The septum will bifurcate the bacteria into two halves, and one copy of the DNA will go to both the chamber, and finally they will get litters. This is the most simplest way of reproduction. So bacteria reproduce the transverse binary fission. So a culture media, if it contain all the essential and non-essential nutrients in proper amount, then they will grow and multiply, and they will form a colony. So one single colony of bacteria are descended from a single bacterium. That's why we call them clone of a bacteria. We call it a colony. Okay. So if you go through the requirements, then you'll find the bacteriological media, basically, it must contain water. It must contain an ingredient called as the peptone. Peptone is the peptic digest of animal tissue. But let me remind you, nowadays there is an growing demand and inclination towards non-animal source of protein, and this peptone powder can also be prepared from some vegetable sources like soybean and some other plant material. So whatever is there, but this we traditionally discuss that these are the component, basic component of culture media. So peptone is a very rich source of amino acid for the organism. And this all the different essential amino acids in appropriate concentration is present in the peptone. It is available in the powder form. And next is this beef extract. For quite a long time, people are using this lean beef boiled and water part to contain all the proteins and minerals. Then it contains carbohydrate, which are transformed into powdered form, and it become a source of culture media. As I said, these days again, this particular component, because of the problem associated with prion infections, we'll be discussing at a certain point these prion diseases. There is a growing demand that the media should not contain any animal components. So it is again replaced with some other vegetable source, but still majority of the media, you can see this is a component beef extract, which is a very rich source of nitrogens, a source of proteins, and also source of lipids, carbohydrate, and it is also a source of minerals, etc. Then the next component is yeast extract. As you know, the yeast are some unicellular fungi, which are, as all of you commonly know, that the application of yeasts are used for fermenting process in beer industry, alcohol industry, or even fermentation of breads. This yeast are used. So these yeast are grown in bulk, and then they are dried to powder form, which is considered to be a very, very rich source of vital amines for the microorganisms. So yeast extract powders are also a component of bacterial culture media. Okay. Then the next component is agar. Agar is not a nutrient. In earlier class also probably I mentioned about this point. You know, this agar is simply a solidifying agent. So agar is an extract of a marine algae called as Xylidium corneum. I guess I discussed this part in another class, but still we can shortly discuss about it. So for ancient time in Japan, the people are using this agar, Xylidium corneum extract, as a solidifying agent. They use it in day-to-day preparation of jam and jellies. And many other activities. They use this agar component. And then later on, it was an accidental thing how this incorporation of agar that took place. As probably you can remember the story of Louis Pasteur as well as the Robert Koch. Yeah, Robert Koch was working on cultivation of bacteria, and at the time they use bovine gelatin as a source of solidifying agent, but that creates some problems because there are certain bacteria which can produce gelatinase enzyme and can cause a liquefaction of gelatin. So a solid culture media become liquefied once the bacteria grow and multiply. So to overcome that, it was an incorporation of agar by Fanny Hesse. As you know, Fanny Hesse, the wife of one of the co-workers with Robert Koch, and she suggested, why don't you use agar? I'm using it daily basis on preparing jam and jelly, and it was so fantastic. And really it worked so nicely, and till now we are using the agar powder as a solidifying agent. Here, just to remember one point, that agar, although it is a carbohydrate in nature, but bacteria cannot utilize agar as a source of energy. So agar is not a nutrient. Please remember, agar is simply a solidifying agent that we use it. And the property of agar is that whenever you dissolve the agar powder in water and when you boil it, agar melts above 94 degrees centigrade, and when it cools down below 45 degrees centigrade, the agar starts solidifying. And when the agar gets solidified, it gives a jelly-like consistency depending on the concentration of agar present there. Jelly, I can explain you, all of like jam and jelly, everyone knows it. Even some gel you apply in your hair for straightening something purpose, and then that semi-solid consistency, that type of consistency is given when you apply agar into the culture media. So as a result, it become an excellent solidifying agent that we can use it in bacteriological media for isolation of bacteria. As you know, the pure culture is possible only in the solid culture media, not in the liquid. Now, if you remove this agar, if you do not add agar into the media, the media will become liquid. And liquid media, in our language, we call it as a broth culture. So we grow the pure culture of bacteria. If you need it for toxin extractions or to grow in the large amount, we use the broth without agar. But whenever we go for pure culture of bacteria, always we use solid culture media containing agar. Okay. Then lastly, it is the to manage or maintain the osmotic balance, sodium chlorides are used to the culture media. So these are the most basic component-wise available with us. Utilizing it, we can design a bacteriological media, and most of the media are well designed for all organisms. Like a particular media, it should contain peptones, say 5 gram in 100 ml, beef extract 1 gram, say yeast extract 1 gram. Likewise, different formulations are already available, and simply we use those formulas for preparation of bacteriological media for growth of bacteria. So please remember, these are the most basic component, and thereafter, depending on the requirements, like if a single kind of media, if you use it, like you'll not be satisfied. All the different kinds of bacteria will not be satisfied. So by incorporating some additional components, some are supporting, some are again inhibiting in different concept. Different type of culture media were developed, and as on today's days, even I can exactly tell you how many different types of media are available, but it will be in thousands. Okay, so it's obviously it is become extremely difficult to remember these different types of media. If you go for catch-all a small classification scheme, that it become very easier for us to understand bacteriological media. So whatever the media available for isolation of bacteria, what I mean about the thousands of media available, this media are belonging to any one of these categories. So for our easy understanding about the concept of different type of bacteriological media, we can classify all the media belonging to any one of this category. In fact, there are certain media which again fulfill the criteria into two different types here. So first,
Sir,
yes.
it seems some are still waiting.
Usually it's not happen. Why? It's I keep it auto admission, and then still why they are not able to, I don't know. Okay, let me quickly enroll them.
Thank you, sir.
But only 29 of you have joined it. I don't know why it's so. Okay. Should I continue? Yeah. Can you see the slide?
Yes, sir.
Okay, fine. So this probably we discussed in one of the theory class for synthetic, non-synthetic media, simple, routine media, enriched media, enrichment, selective, differential. Should I go for again explaining all this again, or I guess we discussed these things in one of the theory class. Okay, quickly, if you feel more elaboration is needed, please ping me and call me, and then I can again explain it. Simple is a routine ordinary media which support the growth of all type of organism, and then in this media, when you incorporate additional nutrient in the form of animal serum or blood, then it becomes an enriched media. Fastidious bacteria, they like this enriched media for the growth and multiplication. Beside this, we had another type of media called as enrichment media, which is available in the form of broth. If your clinical specimen contain very few number of organisms, using it, you can amplify them into a higher number, and then you can grow and multiply. Selective is another kind of media which selectively support the growth of one kind of bacteria and suppressing other. So as you know, the bacteria, they remain in mixed population in the nature. So if you know the selective requirement of your bacteria, then you can design a selective media, and that can be used for their growth and multiplication. Similarly, there is a media it's called differential media. This is the most widely used media which can differentiate the bacteria on the basis of their cultural characteristics. Now let us go through some of the common media that we use it. The most basic media that we use it for isolation of bacteria is nutrient broth. This is a liquid culture, as you can see. It doesn't have any agar. It's simply a peptone in 1 liter of water and the sodium chloride 5 gram. That's the composition of nutrient broth. And this is, it's a basal media. It can support the growth of all kind of organisms, and usually for storage of bacteria, and then for some antibiotic sensitivity test. We propagate the organism here. We never expect a very good luxuriant growth of bacteria in this particular media. So it is, that's why called the basal media. It's contain only bacteria, no other components is present. Okay. So the same nutrient broth, when you add agar, then it becomes a solid culture media. This is an agar plate, and this is solid as compared to the previous broth on liquid one. When you add agar, the percentage of 2 to 2.5%, then it will give rise to a solid culture media, which we prepared in this kind of battery dishes. And this nutrient agar media are most commonly used for antibiotic sensitivity test. And suppose if you isolate the bacteria in the pure form, this pure culture we can subculture into this nutrient agar and keep it for 1 week or 2 week time. So all those basic purpose we use the nutrient agar. Let me tell you, like fastidious organisms doesn't like this media very much. They will grow, will be very, very poor in this media because this is a basal media. So this nutrient agar, basically we prepared in the form of a slant. This is a slant. You can see this. We pour this molten agar and allow it to solidify in a slanting position, and it gives rise to a slope here. And in this particular slope, a pure organism, we can inoculate it, and thereby we can preserve them. This is a method of preservation of bacteria for, say, at least a month or two, for short-time preservation of a bacteria. Usually what happens if we encounter some disease outbreak and we isolate the bacteria, all the study we cannot complete at a stretch. So for that we need to preserve this pure culture for certain period. So for that we use this kind of nutrient agar slant. This is called as the slant, S-L-A-N-T, slant, and use it for preservation of bacteria. So for those purposes we use this nutrient agar. Now here it comes the blood agar. I mean, when you add the blood, animal blood to nutrient agar and that, or the blood agar basal media, then it will give rise to a blood agar media. Blood agar media is an excellent media because the basal media is fortified or enriched with animal blood. So as a result, what will happen? The bacteria will be very, very happy to get the animal protein into their culture media plate, and they will grow like anything. So this is basically for fastidious organisms. So this blood agar we prepare and we use it. So how we prepare the blood agar is that we need a basal media, say nutrient agar, and then there we add 5 to 10% at that particular rate. We use defibrinated or saturated blood. Okay. Now here a problem arises, like the culture media when we prepared, we can sterilize by autoclaving process, but blood we cannot sterilize by autoclaving. We cannot apply heat. So only way is that we have to collect the blood from a healthy animal by jugular puncture in a very clean and sterile environment. I could manage a video from YouTube. Let us see. Please tell me whether you can see the video or not. Can you see the video? Can you see the video?
Yes, sir.
Yes, sir.
Okay, fine. So here, some institute in India, they're showing demonstrations. We need a blood collecting set or even the saline set. We can use it. Also be sterile. Okay. Then we need some scissor to clip the hair. Then alcohol spray, then or iodine solution for decontamination of the skin surface, and the main bottle. This should be a sterile bottle containing some anticoagulant. We can use it. So first we have to. We'll just go a little bit first. We have to clip the hair. Particularly the sieves are preferred for collection of blood in the preparation of blood agar media, but can also be collected from ox. We call the ox blood agar. This area, the jugular vein area, we need to clean it up properly. For that, first we have to clip the hairs, and then we have to disinfect the area very properly so that during collections no contamination occur. So since the blood we cannot sterilize, even by filtration also we cannot sterilize. So only way is to collect the blood very, very aseptically. So after cleaning this area with previously we used the iodine, that's why it's become yellow, and now it's alcohol. We clean up the area in the jugular vein, and then we punch the jugular vein with the needle, and we prepare the apparatus. These are all should be sterile, and then we punch the jugular vein, and the blood will ooze out, and drop by drop it will fall into collecting bottle. So all the stages it should be in the total sterile condition. So likewise the blood can be collected, and the blood wants to come out, and then it will be collecting in the collecting vial. So say 30 to 40 milliliter of blood, we can collect it, and we can take it out, and finally we get this blood like this way. Okay, so this way we can collect the blood, defermented, saturated blood. We can collect it, and then we prepare the nutrient agar 100 ml, and then when the nutrient agar after autoclaving, we can autoclave. We can sterilize this nutrient agar by autoclaving and allow it to cool down to a temperature of around say 50 to 55 degrees centigrade. As you know, at high temperature, if you mix the blood, all the RBCs will be lysed, hemolyzed. So at a temperature between 50 to 55 degrees centigrade, we add this blood collected aseptically from a healthy animal at a percentage of 5 to 10%. And then if you mix it immediately and pour it into the culture plate, you can see the nutrient agar plates are transparent. As you can see it here, it is totally transparent, but once the blood, if you add it, they become opaque. This indicates that this media contained all the RBCs. RBCs are intact. Okay, beside the RBC, it contains the animal body fluids. I mean the blood components, serums and plasma. It containing the different growth factors. As a result, it provides a very conducive environment for the bacteria for their growth and multiplication. They love it as if it is something a party dish containing egg, fish, meat, everything. Okay, they'll be very happy to grow there. They will grow, and this is the scenario where the bacteria will grow into different colonies in the blood agar. So this blood agar also, it is an example of differential media, because this can differentiate hemolytic and non-hemolytic bacteria. Here certain pictures. What happened? The bacteria, if it contains some toxin like hemolysin, most of the bacteria, they harbor, not all. Hemolysins, when they grow, they will release the hemolysin, and this hemolysin will cause lysis of RBC. As a result, what happened when the bacteria will grow surrounding the bacterial colony, all the RBC will get lysed and the media become transparent. This is called as the hemolytic reaction. So in coming days, when we talk about the hemolytic bacteria, we'll be knowing about complete hemolysis, partial hemolysis, alpha hemolysis, beta hemolysis, so many other things we are going to discuss it. Just a glimpse I'm giving it here, how the hemolytic reaction. Just look at this particular slides where you can see this white pigmented bacteria doesn't have any hemolytic activity. Probably they are non-hemolytic, okay? Whereas the other type, the yellow type bacteria, they are causing some sort of clearance surrounding the colonies, so they are considered as hemolytic. So the blood agar is also an example of differential media, because it can differentiate hemolytic and non-hemolytic organism. At the same time, blood agar is also an example, classical example of enriched type of media, because the vessel media is fortified or enriched with animal blood or animal protein. So all organisms they prefer. Okay. Now there is another media, what we call as the chocolate agar media. Let me see anybody again trying to join here. No. Yeah. Fine. So this is an example of chocolate agar. It looks like the chocolate. This is basically the blood agar. When you heat it up at a high temperature at 90 degrees centigrade, or you mix the blood at a high temperature to the molten agar, then all the RBC will get hemolyzed and it will give rise to a chocolate color consistency. There are certain bacteria which prefer chocolate agar rather than a blood agar. So in coming days, we'll be knowing there's an organism called as Hemophilus. Hemophilus Influenza is a very common human pathogen for which all of you have taken your vaccines in your childhood. And in case of the animal, we had Hemophilus parasuis. Nowadays, it is called as Gaseous parasuis bacteria, which, like this chocolate agar, they depend on the two growth factor called as X and V factor. X is the hemine, and V factor is nicotinamide adenine dinucleotide. These two are essentially needed for their growth. So once the blood cells will lyse, all these components will be available to the media where they can grow and multiply. So this is a chocolate agar. So some of this media will be learning this way. Then coming to another, it's called as the brain heart infusion agar, as well as we had the brain heart infusion broth. If it doesn't have the agar, then it will be a broth. So this is a classical example of enriched type of media. It contains very good nutrition from brain tissues, then heart tissues, beside the other common components. So this is this particular media supports the growth of the bacteria very much, and the bacteria, they are very happy to grow in this media. And if you inoculate any bacteria culture into this media, they immediately after 24 hours you can see very luxuriant growth. So when we plan to go for any molecular diagnosis or detection, or if you want to study the toxin produced by the organisms, then we prefer this media to grow the bacteria, so that all the virulence factor they express to the highest amount, getting all the nutrient in the proper concentration. So this is brain heart infusion agar. As you can see, the agar is 15 to 20 grams. That means 1.5 to 2%. If you remove this agar, then it will be a broth, brain heart infusion broth. So it's another example of enriched media. That come to a media called as brilliant green agar. These are some very commonly used, and this media, very commonly we use it for isolation of salmonella, the typhoid causing bacteria. Whenever we suffer from typhoid infections, the laboratory days to grow the organisms in this kind of selective media. So brilliant green agar, it is again a selective media for isolation of typhoid organism. So it contains the most basic like peptone, yeast extracts, and then it contains agar. Beside this, it contains lactose. Sugar is added. 1% of sugar is added, and then it contains sodium chloride, okay, sucrose, lactose and sucrose, two different sugar and two different indicator. These are pH indicator. One is phenol red, the other is brilliant green. Because of the presence of this indicator, what happened? The organism can be identified. If it become alkaline, if the bacteria they can grow and multiply, cannot ferment this sugar, then some alkaline reaction are produced, which will be indicated with phenol red. And if become acidic by fermentation of the sugar, then it will give a bright green color. So at the same time, it can be used as a differential media. Okay, so let's see how it happened here. Okay, this is a normal uninoculated plate of brilliant green agar. Okay, and then here these organisms, this is the Escherichia coli, E. coli organism, which produces acidic reactions. So they can ferment this sugar present here in the media, lactose and the sucrose, and then because of sugar fermentation, a lot of acids are produced, and the green color appears, acidic reaction appears into the culture plate. So looking at it, we can say that this is an acidic reaction produced by this organism. At the same time, the same media played when the salmonella organism that causes the typhoid disease, enteric typhoid disease. This organism cannot ferment any one of these sugar, lactose and the sucrose. They cannot ferment, so they cannot produce any acid. Rather, utilization of protein, there will be production of alkaline reaction. So when it becomes alkaline, the pH of the media become alkaline. You can see the color transform into bright pink color. So this is an indicator media, or this is a differential media which can differentiate acid production, alkali production. So salmonella typically produces this kind of discoloration, dark pink discoloration of the media. So obviously it will help us in identification. Suppose you inoculate some still sample into this BZA and you could notice pinkish discoloration of the media, then we can suspect of presence of salmonella. Okay, if it is greenish discoloration, then obviously we can say it doesn't have any salmonella organism because this is acidic reaction. So thereby this kind of media, we can call it as a differential media, as well as we can call it a selective media for isolation of bacteria. Then another media, which is another very frequently used laboratory media, is called as the leucine methylene blue, but having a very specific purpose, this particular media, and we use it for isolation of Escherichia coli bacteria. Leucine methylene blue, these are two dye bacterial stain on leucine and methylene blue, mix up with the basal media. So leucine Y and then methylene blue is added. So because of which what happened, this Escherichia coli bacteria, they typically produces a metallic scene colonies. Just see, this is an very luxuriant growth of Escherichia coli. They can cause oxidation of leucine, and the whole colony appear as a metallic scene, as if a drop of molten gold, you put it in a black surface. So this kind of metallic scene colonies are produced, and exclusively only few organisms they produce, and E. coli they persistently produce this kind of changes. So as a result, if you want to isolate this Escherichia coli from urine sample or any other stool sample, straight away you can inoculate into the leucine methylene blue agar. And then if any colonies are signing like this, we can say that this might be almost surety, we can say this is Escherichia coli. You can see this is another, this is no metallic scene produced by Enterobacter aerogenes, and here it is the metallic scene produced by Escherichia coli. So this kind of media help us in identification and in the process of disease diagnosis. Then coming to another media, it's Macron's lactose bile salt agar. Shortly, sometimes we call it the MLA, Macron's lactose agar. So this is an example of, again, differential media which can differentiate lactose fermenting and lactose non-fermenting bacteria, and are very, very commonly use it for any diarrhenic pathogen identification. So it can differentiate the important members of Enterobacteriaceae. And first thing is that this is again an example of selective media. Why? Because this media contain bile salt, this component bile salt. Now you know this in our digestive tract, in the duodenum, from the liver, the bile that comes and they pour biles. So you won't get the bile in the gastric mucosa or in the nasal surface or in the mouth cavity. We never get any bile salt. So biles are mostly present from the duodenum onwards. So the bacteria that are present in our intestines, they are always bathed in bile. All the time they get exposed to the bile and they are tolerant to the bile salt. But think about the bacteria which is present in our nasal cavity, in our skin, in our lungs, or in the trachea, or those organisms which are never exposed to the bile. So bile is toxic for them. So this media contains the bile salt, so that only the bacteria which can tolerate bile can grow. The bacteria, those which cannot tolerate bile, cannot grow and multiply. So because of this particular component, presence of this component, this is also can be called it as a selective media, because they will suppress the growth of bile sensitive organism. Only the bile resistant organism can grow and multiply. At the same time, again, it contains the lactose sugar, and the lactose fermentation can be detected with the help of the pH indicator. It is the neutral rate. Okay, at the neutral pH, the color of the media become light red color media. But when lactose fermentation will be there, when more acid formation will be there, the intensity of red color increases. And when it becomes more alkaline, means towards the pH 8, 9, like that way, the whole colony will become pale color. They will lose the color because of this presence of neutral red indicator. So it can differentiate this lactose fermenting and non-fermenting bacteria. So lactose fermenting bacteria, they produces intense red color colony, whereas lactose non-fermenting bacteria produces pale color colony. Let us see a process here. This is an McConkey's lactose agar, and one half of the agar, this lactose fermenting bacteria were inoculated, the other half non-fermenting. Now you can see the difference. So this indicates they are able to, this group of bacteria are able to ferment the lactose, and there is production of heavy amount of acid here, and the neutral lead is appearing in intense red color. On the other hand, this side, the bacteria, they cannot ferment the lactose present in the media. As a result, they are producing alkaline reactions. The pH indicator will give pale color, okay, or no color reaction. So it can differentiate the lactose fermenting and lactose non-fermenting bacteria. So here, let's take a short gap and then see this video that I prepared it in today morning. How we can prepare McConkey lactose agar. This is the video in our busy lab. We prepared it. Sorry, this is the McConkey's lactose agar. So these are the compositions. Already I have shown it in the slide. Additionally, it is having the crystal violet, which acts as an inhibitor for gram-positive bacteria. And in all this media bottle, they is to indicate how much gram. These are all ready-made media containing all the individual ingredient in powder form as a mixture, and the indication is given like 45.53 grams you need to mix up in 1 liter of solution, and then you can get the media. It also contains the agar. We need the media. Then this is the water, distilled water. We use it. Then we need the electronic balance, and then as per the requirements, we can measure up the quantity of media. We tear the balance, and then can you see the slide video?
Yes, sir.
Okay, fine. Now he's just measuring as per the requirement for the volume, and then actually we did it twice. Initially we did it here. Again, we are going to increase the volume of media. So this way, this dehydrated media, this contains all the peptone, beef extract, then it contains yeast extract, it contains agar, it contains this bile salt containing lactose, even the crystal violet, etc. So we have to take the actual measurement, and then we'll put it inside a conical flask. Then we'll take it to the laminar flow for adding the water inside the laminar flow. We'll pour the water. We'll mix up this media properly. So once this media we dissolve into the water, we need to check the pH with the help of the pH paper here descripted. all the media, they are having a definite pH, adjusted pH. So as per the company's recommendation, when you measure it properly, then obviously you will get the receive the same pH. So we allow it to dissolve. Then we will check the pH of the media. These all things you're supposed to do in your practical. So once you'll be returning back, we'll do all the things and the demonstration will be given to you. Let us take another cap here. Yeah, part B. Then we are going for preparation of another very special media. It's called as the Robertson cooked meat broth. It's not this video. Okay, once we'll check everything, then we keep it in the. This is another media. The video is missing here. Okay, then this is the autoclave. We switch on the autoclave and then close it, and then you can remember the autoclaving temperature and the time. It's the 121 degrees centigrade for 15 minutes. And since there is no any temperature marking here, it is adjusted with the help of the pressure gauge. We know at 15-pound pressure, the temperature achieved is 121 degrees centigrade. So this is the pressure gauge. And once this pressure gauge reaches to the 15-pound, this particular area, the internal pressure will increase. Then from this point onward, we'll count 15 minutes time. And we'll hold it for 15 minutes at 15 pounds. That will achieve the autoclaving process. Okay. So thereby we can get the sterile media. Okay. So once we get the sterile media, then it is our part to prepare the this is the flux after autoclaving. It's a lot of it is quite warm. So we clean up the laminar flow area. We'll switch on the flame, and then these are the Petri plated disposable Petri dishes where we'll prepare it, the culture media. All this practically you're supposed to do in your regular class. So you take out everything. So even some test tube to preparation of slant, I'll show you. If you have any question in between, you can ask. So we'll switch on the flame. Already airflow is here, but still we'll not take any risk. And this whole process should be, we have to do it very, very aseptically. It means no contaminations, no scope of any external contamination. So these are all sterile plate. If I ask you how these plastic plates are sterilized, your answer will be by gamma radiation. And these are some sterile plate individually packed. He's Dr. He's Sarat Singh, our FCLA in our department. So he's giving the demonstration. So after autoclaving, we have to open it near the flame inside the laminar flow only. You have to be careful. You should not open it outside this laminar flow. Everything will be contaminated. Then we'll pour this media. Now it is quite warm. That's why the agar is not solidifying. So temperature is more than 55 degrees centigrade. That's why it is still in molten condition. But once it will cool down, we'll keep the lid a little bit open so that the excess moisture will evaporate from the Petri dishes and no water clogging will take place. So thereby we prepare the culture plate. So yeah. So once this plate will get solidified, then we have to keep this plate overnight in inverted position. We have to keep it in the incubator overnight, so as to make sure that all processes are aseptic. Tomorrow, if we do not get any growth of bacteria, then we say that our media is sterile. So this is a process of preparing bacterial slant. As you have seen in the nutrient agar slant, this is Macomphis lactose agar slant. Close it and allow it to solidify in the slanting position like this way. Okay. Once the agar will solidify, it will give to a slant. Okay. This is our Macomphis lactose agar, and this is the slides you can see already we studied. Then this is the Robertson cooked meat broth. This is a special media that we use it for isolation of anaerobic bacteria, Clostridial bacteria. So basically, it is a meat particle, cooked meat particle as a media. It contains the peptones and other broth, and then it's a meat particle, as you can see it here. And the media bottle also, you can see this meat particle. I think one video is here. This is another process. We take a beaker. We need the distilled water, and then the media. So this is the media bottle. Robertson cooked meat broth. Cooked meat broth. RC means Robertson cooked meat broth. So the positions are given here, and it is showing 12.5 gram in total 100 ml. So accordingly, we need to calculate, and we have to mix this meat particle with water. Okay, the same process. In this balance, we'll tear it, and then we'll measure this meat particle. So this media is specific for isolation of Clostridial group of bacteria like Clostridium tetani, Clostridium joviae, those anaerobic bacteria. So this media are also called as the reducing type of media, which can avoid incorporation with air. So we'll talk about the reducing media the later part. So likewise, this meat particle, we measure it. And then we mix up with the recommended volume of water. Can you see this video? Is it visible?
Yes, sir.
Okay. So we mix up. This all practically you're supposed to do. Okay, fine. Then yes, repetition. After that, we dispense this meat particle into the individual tube, and then we autoclave it. And this media, we use it for anaerobic isolation. This is in short about the different type of common media, although there are many other media we use it. I hope that this will give you a glimpse about different types of media, and then how to prepare it. Then next important point is how we can inoculate and get a pure culture of bacteria. So if you go to the method of inoculation of bacteria in the laboratory media, basically, although there are 4 or 5 methods we describe, but most commonly used method is the strict plate method. Okay. Here, what we do physically, we spread the bacteria on the surface of the culture plate, and physically we separate them with the help of inoculation loop and allow them to incubate. And when the bacteria will transform into a colony, visible colony, we can carefully pick up and get the pure culture. So another method is also called a spore plate method. Some laboratory they prefer this method also, but spore plates are basically used for counting of bacterial load into a specimen. But for isolation purpose, it is most commonly used method is a strict plate method. Okay, so how basically it happened is that this is a sterile culture media plate. We pick up the samples, and then what we do first, we smear this loop full of culture at one corner of a sterile plate on the surface only. Remember, this is on the surface of the agar media. This is called as the pool, first pool. Then from the pool, we'll make some streaking line touching the pool to away from it. This is the first, the second streaking line. Then we go for another directions, streaking line, then next streaking line. So that what happens, suppose this is the pool, after incubation, this will be the picture. In the pool, so many number of bacteria will be there that all colonies are clumped together. You cannot visualize a single colony here. And when you make this streaking line like this way, when you make this streaking line, physically you're separating the bacteria in this direction. Then again in this direction, and then finally in this direction. What will happening from this initial high concentrated population, gradually you are diluting the bacteria to the other part of the plate. This is what we call the streak plate method. You're simply streaking it, just like a pen writing in a paper. You are making some streak line using the inoculation loop, and finally, in the final streaking line, this particular streaking line, one bacteria fall down at certain places, and when you incubate, one bacteria give rise to one colony. Now, suppose your sample contain variety, different types of bacteria, then finally, you can see if the sample contains 4 different type of bacteria, you're supposed to get 4 different type of colony in this culture plate. So as you know, the bacteria, they differ in their shape and size of the colony. So we can get this all the different type of bacterial colony from the sample, and then using an inoculation loop, just concentrate this colony. You just pick up, touch this colony. This colony, all the bacteria, they are descended from a single bacterium. So this is a pure culture. So you can pick it up and put it in another culture plate. So you will get a pure colony. In the culture plate, all the bacteria will be of same type. Similarly, if you can spot 4 or 5 different colonies and individually pick up the colonies and put it in subculture into another 4 plates, you will get the pure colony. So this is the method called as the streak plate method. Very, very simple. All of you will be doing this particular practical. I will give you a demonstration here. We clean up the area with alcohol and the spirit. We took the flame here, and then we'll mark it. This is the stool specimen we have received today in our laboratory. We'll give the same specimen identity to the broth culture. This is the broth culture. Okay, and at the same time, so we'll take a sterile MLA plate, as because it is a stool sample. We'll prefer the Macconki's lactose agar. It was prepared yesterday, and then we'll give the same marking as the E13, and we'll write today's date. Today 26 again, yeah. Then we'll just give you demonstrations. We'll take an inoculation loop, and then first we'll sterilize it. This is called the red hot sterilizations. When you flame it, the whole inoculation will become red hot. So the temperature reaches almost 300 degrees centigrade. This is a nichrome wire. So all any bad type of organism, its presence, they will burn out. Okay. So we have to cool the inoculation loop by keeping few second time. So under no circumstances, the hot loop we should straightway touch to the clinical specimen. So we have to cool it by touching to the wall of the tube, and then gradually we can touch the sample. Okay. So by touching the samples, we'll take out the loop full of culture, and then we can mix up with the broth. This is sterile broth. So we just mix up or inoculate the clinical specimen. So whatever the bacteria will be present in the stool specimen, they will grow in the broth overnight. And tomorrow we can explore it, how many different type of bacteria is present there. So at the same time, I'll just give you a demonstration how this stool sample can directly be inoculated into the culture plate. And we're expecting some growth tomorrow. So we'll cool the inoculation loop, and then we'll take a specimen. Then we'll take the culture plate and we'll go for streak plate method. So at one corner, since it doesn't have a corner to the periphery somewhere, we'll just smear it on the surface of the agar. Remember, it is on the surface. This is called as the pool. So starting from the pool, we'll make some streaking line. 1, 2, 3, 4, 5, like that way we'll make some streaking line. Then we'll turn the plate from the first streaking line again. 1, 2, 3, 4, 5. This is the physical method of separating the bacteria from the pool. 1, 2, 3, 4. Then we'll briefly burn it to reduce the load of bacteria if it is present. And we'll go for the final streaking. We'll cool it. Then we'll make the final streaking. Okay. So this is what we call as a streak plate method. So by this method, what you can do physically, you can separate the bacteria from the pool to the other part of the plate, so that in the final streaking line, the individual bacteria, they remain 1, and then can be transformed to colonies. So when the incubator will just keep the broth and the culture plate, and then we'll incubate it. And as you know, these are mesophilic bacteria. At 37 degrees centigrade, we adjusted the incubator, so we incubate it. So this is something that I prepared. By streaking plate like this way, I can see varieties: smaller, bigger, and bigger size, different size of bacterial colony. You can see that help us in identifications. And very carefully, when you pick up one colony and put it in another, you'll get a pure culture. Sometimes 2, 3 times we need to streak it to get the final pure culture of bacteria. So this is the way how we can go for isolation of bacteria. Now, the last part. Think about the anaerobic bacteria. As I said, that there are certain bacteria which cannot tolerate even the slightest concentration of oxygen. So for those organisms, we need to provide an environment which doesn't have any oxygen. So it is very well possible with minimal cost in a particular equipment, what we call as the anaerobic jar. So this particular device is called as the anaerobic jar. Okay. So sometimes also called as Macintosh filled anaerobic jar, developed by the scientist, and nowadays it has been modified from available. So this is a simple chamber which you can make the lid airtight by some devices. So what we do, we keep this inoculated culture plate inside this chamber, and then we put a gas pack system. Gas pack systems are certain salt mixture. These are some chemical reactions which absorb the atmospheric oxygen. When this salt reaction will start, they will absorb the atmospheric oxygen, and carbon dioxide or sometimes hydrogen gases form inside this chamber, and some water accumulation takes place at the point, but the oxygens are totally removed from this particular chamber. And this kind of device, we call it as an anaerobic gas pack system. Okay, these are certain salt mixture. We are not going for the details what type of chemical reactions occur. Mainly, we'll talk about the applications. So when you open up this kind of airtight packet inside this container, then they will absorb the oxygen and anaerobic condition will prevail. After that, this whole device we can put it inside incubator. So if you don't have this kind of anaerobic gas pack system, another, I'm just showing it here. This is a device in our lab. These are airtight chamber. We'll open up the lid. Okay. So there is a process what we call as the inverted candlelight method. This is absolutely zero cost process where you can create micro aerophilic condition, not total anaerobic. We can create micro aerophilic condition. So for that, we can place the petri plates inside this canister, and then we can light a candle. This is, of course, not total anaerobic. This is microaerophilic. As you know, there are many bacteria which prefers in reduced oxygen tension, carbon dioxide percentage of 5 to 10. They prefer it. So this whole things will put it inside the anaerobic gas jar. Okay. Then what we'll do, we'll close this lid tightly. Okay. So we'll close it and then we'll screw it tight, and so that no air leaks. So what will happen? This candle will burn all the oxygen inside this chamber, and that carbon dioxide will be released. So what will happen? The oxygens will be removed, but this process cannot make you assurance that total removal of oxygen. That's why we call it as a microaerophilic condition. You can purchase. So long the oxygens will be there, it will burn, and after that it will off. Okay, maybe the next video to go. Yeah. Is it this one? Yeah. So what will happen once the oxygens will be totally burn off? You can see that gradually the candle flame is also off. So what happened? This oxygen part is totally removed and is replaced with carbon dioxide. So inside this chamber is now a microaerophilic condition is prevailing. Okay, so this jar after microaerophilic condition, we can keep it inside an incubator for temperature. So the more improvised, if you want to make assurance of total anaerobic condition, we need this kind of anaerobic gas pack system. So this is for 3.5 liters, exactly our container size. So inside this packet contain salt mixture. This is a salt mixture. This is already used one. I have not used a new one. And there is an indicator tablet which can indicate the aerobic and anaerobic condition. It is basically remain pink color, but once anaerobic condition prevails, it will lose its color. This is some sort of oxidation reduction reaction, and reusable tablets are there. So what we do, we place the plate after inoculation, and this packet, immediately after opening from this packet, we have to fit it. This is an already used one. Then this whole things, we put it inside this chamber. Okay, this is an anaerobic jar. Then close the lid. So this way, if you use an anaerobic gas pack system, it can assure total removal of oxygen. Or strictly anaerobic organisms, we grow it by creating these conditions by anaerobic gas pack system. So this way we can take care about the anaerobic bacteria, and then we can grow them. Okay, so whenever the organism, you know, it is a strictly anaerobic, then you have to take care, otherwise the bacteria will never grow and you won't get in any colonies. Okay, whereas there are many organisms which are microaerophilic. When the atmospheric condition is filled up with 5 to 10% of carbon dioxide, then they are happy to grow there. So by inverted candlelight method, we can create that condition, and we can incubate. So microaerophilic bacteria, if you try to grow in the total aerobic conditions, then you will not get the optimum growth. So not only the nutrient content, but also the gaseous requirement is quite important for isolation of bacteria. Okay, so thereby in this class, I tried to make you give you some information about the bacteriological media, how it can be prepared, and what are the basic components and the different categories, and some of the example that is given in your manual. Exactly, I depicted here. At this stage, of course, it will be difficult for you to remember the function of each and every media, but at least you will get an idea that how these bacteriological media help us in identification and isolation of bacteria. So in a later part, when we specifically discuss about the disease like staphylococcal disease, streptococcal disease, bacillus anthracis infections, then classical infection, then one by one we'll again discuss about the isolation, specific isolation process and identification process of those bacteria. So I guess this particular lecture will provide you some information about the isolation process that bacteria that we carry out in our laboratory on a regular basis. So some components you need to do it by yourself once the normalcy comes. So today's class I prepared up to this. The next class will talk about some of this area again and identification process. So after that, we'll be shifting our practical towards the immunology practicals. Okay. So thank you very much. 23, 24.